Good evening, all. Welcome to the penultimate talk this term at the Oxford University Scientific Society. I am Luna, and I'll be your host for tonight's event. Today, we have Nicholas coming to talk to us about he and his group's work on rapid viral testing. Nicholas is a DPhil student in Professor Kapanidis' laboratory at the Oxford University Department of Physics. If you have any questions for Nicholas, please write them in the comments section as the talk proceeds. We're pleased you can come to join us tonight, Nicholas. The stage is all yours.
Hi, everyone, and thank you for the invite. I'm really excited to be here. So, can everyone see my screen? I assume so. So today I'm going to talk to you about how you can detect viruses in under 5 minutes. This is a work I first started working on during my master's project. It's a during the fourth year undergraduate course here in Oxford, and then I stayed to do my PhD, and we got some pretty nice results that I'm going to present. So I would like to start this with a quote from the International Health Regulations Review Committee after they've been asked to assess the level of readiness of the world in case of a future pandemic. So the quote says the world is ill-prepared to respond to a severe influenza pandemic or to any similarly global sustained and threatening public health emergency. And unfortunately for all of us, that came to be true. So we're going through the SARS-CoV-2 pandemic. It needs no introduction. We're currently approaching 1.5 million deaths. And according to the International Monetary Fund, the pandemic will cost the global economy $21 trillion. So we can see the total cases and the total deaths are actually increasing steadily. So we don't seem to be towards the end of the pandemic anytime soon. This is the worrying. Of course, we have some exciting news from the vaccine world, and hopefully they'll be able to be deployed, but there are still many unknowns and whether we will be able to be over the pandemic in the short future. So what is actually SARS-CoV-2? To start with, it stands for Severe Acute Respiratory Syndrome, and it is a virus. It is an enveloped virus, which means it has a lipid membrane with lots of spikes attached to it, which are proteins. The most well-known protein on the surface of SARS-CoV-2 is the spike protein, the S protein, which is about 20 nanometers in length. And it is an RNA virus, which means that inside the lipid membrane, we can find RNA segments. So coronavirus is a pleomorphic virus, like most viruses, which essentially means that it comes in many shapes and sizes. This particular virus begins with 50 to 200 nanometers in diameter. So they are really, really tiny. So viruses have always been a threat and will always be a threat. Since the 1918, we have faced 6 pandemics that have killed millions of people worldwide. It is not a matter of if, but when the next is going to strike. So apart from all these different viruses that became pandemics, we also had to face a few others, such as SARS and MERS and Ebola, that have devastated local economies and had the potential to become pandemics. So it is only a guess game whether the new pandemic is going to be a flu pandemic or another coronaviruses, but it is coming. But it's not just all about pandemics. Infectious diseases actually account for 20% of the global mortality. And every year, we have flu epidemics, seasonal flu epidemics, that affect about 9% of the world's population annually. It is particularly hard to differentiate between different viruses just because they have very similar symptoms. For example, if someone has a fever and a cough, this is very common symptoms for many viruses. So that's why rapid, accurate diagnostics are essential. More specifically, coronaviruses are essential. And we can actually see this in a scale of a pandemic when lockdowns had to be implemented around the world. We are currently going through a second wave. US has more than 2,000 positive corona cases a day. And these are some photos from Core Market Street. Usually it's a very busy place, but this was taken during the first lockdown. It was empty. So it has changed the way we live, and our normal now is very different from what it used to be. So lockdowns can be effective if implemented properly, but they also carry devastating consequences for the economy and the mental health of individuals. With mass testing, which is actually the moonshot pledge of the government to be delivered by February, the aim is for individuals to be able to be tested regularly. Thus, only people that actually have the virus will need to isolate, and not whole counties or the whole country. So essentially, the more tests, the more information about the spread of the virus we have, and of course, the better decisions we can make in terms of minimizing the spread and saving lives. So a quick identification and not just waiting for people to develop symptoms can also help prevent the spread in a more proactive way. Essentially what is happening now, if someone has the symptoms, they have to take a test, and they sort of, but in that period of time between developing the symptoms, they could have been spreading the disease, which is quite worrying and is contributing to the problem. So there are many detection methods for viruses, and many more have hit the market since the pandemic has started. So we have a few ways to detect viruses. One of them is electron microscopy. This is not used for regular testing, but it is a way to visualize in the sample what the virus looks like. It's very detailed, but it takes hours, and it requires very expensive equipment and trained people to carry out the procedure. We can also grow the virus in the lab, but this is a very time-consuming procedure which takes days or weeks, but it is very highly accurate. More common tests that have been recently adopted by the Oxford University are lateral flow assays that can be either antigen-based or antibody-based tests. These are marketed to be a lot faster. They don't take hours. They take around 15 to 30 minutes, but they tend to suffer in sensitivity. So the sensitivity can range from 40% to 70%. So they're not accurate enough. And then we have the gold standard, the RT-PCR, which essentially amplifies the genome inside the virus, and it can provide very accurate results. The downside is that it needs to be done in a lab by trained professionals, and it takes hours. So just to point out that antigen tests tells you if you have the virus right now, and antibody tests might tell you if you had the virus in the past. So they're not actually useful at telling you whether you have the virus right now or whether you may be spreading the disease. So we can say that all the current detection methods come with pros and cons, but none of them have the potential for a mass testing implementation, partly due to the cost and, most importantly, the time it takes for a test to be carried out. So even rapid tests, lateral flow tests, take 15 to 30 minutes, but they're also single use, and it's easy to get a false negative as they suffer from low accuracies. So I've sort of tried to group together what in general are the cons of the current detection methods, which essentially most of them require the lab, they're time consuming, There may be some supply issues because they do require, some of them, expensive reagents, and they compete with each other for those reagents as well. Some have low accuracy, whereas others are expensive, and they normally tend to detect only one particular virus as they require specific reagents. So what are we proposing? I tried to make it easier to present by separating our method into 4 main steps that I will go into more detail in the next few slides. So essentially what we do, we label virus particles, we image them under a microscope, then we get the resulting images and we process them, and then we feed the result of that processing into a machine learning algorithm that is able to distinguish different viruses. So let's dive right into it. So to start with, we have the labeling. The labeling was actually invented in the lab by Professor Kapanidis, Dr. Nicole Robb, who is now an assistant professor at Warwick, and Johnny Taylor, who was then a master's student in physics, and he's now doing a PhD at Imperial. So the basic idea is that because of this lipid membrane of the virus, as we've talked about before, the viruses have a net negative charge. We achieve the labeling with a labeling solution which contains calcium chloride. Calcium is a divalent cation, which means that it has a +2 positive charge, and single-stranded DNA, which also has a net negative charge because of its phosphate groups. So essentially what's happening there, we have 2 negative things, 1 positive thing. So essentially the calcium cation mediates the interaction, acts as a glue, and thus we achieve labeling. It's also worth mentioning that the single-stranded DNA also comes with a dye attached to it, a photoactivatable dye that essentially emits light under a laser. So this is a very simple method for labeling things. It is unspecific. It's not specific to any virus, so it can label anything with a membrane. So any enveloped virus, and it's instantaneous. And also because it's just calcium chloride with some DNA, it's also very cheap. The next slide is going to be me to demonstrate how just performing the experiment, just showing some diffusing viruses to show you how fast the labeling works and that it is indeed instantaneous. With our labeling methodology, we can label the viruses instantaneously. So we begin with adding a small part of the labeling solution into the tube and then mixing it with a small part of the clinical sample. Then we immediately transfer the solution into the well, and you can straight away see the beautifully labeled virus particles. So this is me in the JR, and this is what I spend most of my time doing nowadays. I'm trying to process clinical samples of viruses, and under the microscope, that's what they look like. They look like bright blobs diffusing around. You can tell that they are indeed viruses because of their size and because of the way they're diffusing. We can easily calculate how we expect them to be diffusing based on their size and the temperature and the viscosity of the solution. So as we have seen, the labeling happens remarkably fast, and it is indeed instantaneous, and it's as simple as it looks. You have a clinical sample that contains some virus, and you just add some labeling solution, and the interaction is instantaneous. So the next step is the imaging. For the imaging, we actually stick the virus down to a slide to make sure everything is in focus. When things are diffusing, you get some particles that are in focus, but most of the things are not in focus. And we use a technique called total internal reflection microscopy, which essentially means we shine the light such that it just doesn't go through the sample. So essentially you get an evanescent wave, and that's enough to excite the dyes and they emit light. So we're using a microscope that has two lasers, red and green, and the single-stranded DNA, we use two different ones that also have a red dye and a green dye, for reasons that will become obvious in the next step. And therefore we are able to capture images. So I'm going to show you, this is a GIF I made from some data in the lab. So this is what the virus particles look like when they're stuck down. It's just really bright blobs. You can easily distinguish them from the background because they're so bright. And then we're using a high-end microscope, which was actually first invented in the lab by Professor Shiailis again, and now is a commercial company. And this has the capability of rapidly scanning through the sample and taking hundreds of images just within a minute. So the next thing we do once we have the images, we have to pre-process them to get them ready for the machine learning algorithm. So we can see on the original image how it looks like, and it looked like just how it's meant to look like on the microscope as well. And then what we've done, we actually converted it through some filters. It went through some filters. Adaptive filtering is called. We were able to turn the image to just black and white. So the bright blobs have the maximum value. There are only two values, 1 and 0, and the black is the background, is just 0. And then we filter them for size. So you can see that the small stuff is missing. That's because we know what the size of the virus should be, the minimum size of the virus. And we know that after, for example, if you have only something that is 1 pixel, is not big enough to be a virus, so we can discard it. And similarly, it rarely happens, but you also have a maximum value. So if there's something very big, like a cell, it will also get dropped out and not taken into account. Then the next step, because we're using two different dyes, we're able to get images in both the red and the green channel. This is not a real image, it's a reconstructed image of the localizations. And essentially what we do, you can see the red and the green, and they correspond to things that had the corresponding dye. But when you think things that are yellow, that means that they are colocalized particles, which means at the same space you have the red dye and the green dye, and that implies that it has a high probability of being a virus. Because our labeling is unspecific, we expect them to bind randomly, and both of the dyes and the DNAs to bind to a particle, to a virus particle. So what you go and do is, we go to the localizations, and we go to the filter image number 3 and the 6th image, and we draw bounding boxes around the signals on 3, but only for those that are colocalized. So, and then once we have the size of the bounding box, we go back to the original image and we draw a bounding box around the original signals that haven't been processed at all. And the result is these bounding boxes, which are saved individually. And this is what we're using for the machine learning part. So let's imagine we've done all of our experiments, and you want to be able to distinguish between a positive sample, positive being it has a virus, and a negative sample, which means it doesn't have a virus. So we take all these bounding boxes, and just to give you a rough idea, depending on the virus and the quality of the data, we may need anything from 5,000 bounding boxes per class to tens of thousands. So all these images go through a convolutional neural network, which what it does, it tries to extract the most meaningful features, such that in the future, if you try to input an image, it will be able to tell you what it is. So essentially, it goes through all the images in class 1 and class 2, and it tries to find what makes them different, or what things that, or what positive samples look like and what negative samples look like, and is trying to be able to differentiate the two. So once all the cycles, all the epochs are complete, what do we end up with is a trained model, which is actually reusable. So you only need to train once, and once you have your trained model, you don't have to retrain. The last I've tested, the first time I trained a network was back in March, and I've tried one about a week ago. So about 6 months after the first training, it still works. You don't need to retrain. You can still predict if the sample is positive or negative. So with deep learning, it's hard to know exactly what is picking up, but we do know that there are differences in the intensities, the sizes, and the shapes of the viruses, and probably that's some of the features that the algorithm is using. All of them are actually related to the differences in the labeling, partly because they have different sizes, but also because the surface chemistry is different. So in the beginning, when I showed you the cartoon of SARS-CoV-2, they had different spikes. You can have many different spikes, and actually that's what differentiates essentially viruses. So they have different proteins on the surface, and that means that their surface chemistry is different, and therefore the labeling works differently. So just to summarize, in step 1, we label the virus, which is a cation-mediated interaction. It is universal. It works for anything that has a membrane, and it is very cheap. It also works instantaneously. So for step 2, we image the sample. We use a wide field microscope, so you can get a big area for imaging, a big field of view. It is rapid, automated, and because of the instrument you're using, you can use multiple acquisition, which takes seconds or 1 or 2 minutes. So for step 3, we have the image analysis, pre-processing, and getting things ready for the convolutional neural network. It is currently rapid, and as it is automated, so it usually takes seconds unless you want to process tens of thousands of images. So for hundreds of images, thousands of images, it just takes a few seconds. And the last bit is the AI classification. So it's a custom designed convolutional neural network we made that approximately needs 10 minutes to be trained, depending on how many images you're trying to feed. The estimate is for about 20,000 images. And after that, the trained model can give a classification result, whether something is positive or negative, or which virus it is, within a few seconds. So altogether, all this can be done within 1 to 5 minutes. And it's 1 to 5 and not 5 or 2 specifically, because depending on the viral load, that means how many viruses you have in the sample. You may need only a few fields of view, so it will approximately take you just 1 minute, or you may want to scan the whole sample, which will take 5 minutes. So let's just run through an example. So it is of particular interest how a coinfection would work between coronavirus, the SARS-CoV-2, and a different and an additional different virus. This is actually very relevant now because we don't exactly know how SARS-CoV-2 and the seasonal flu strains will interact together in the same host. So you may want to be able to, it is also going to be interesting, we won't know because the symptoms are so similar, whether someone just has the flu or they have SARS-CoV-2. So what you will do in this case, if we're trying to implement this, is get a lot of samples that we know, that they're SARS-CoV-2, image them. Let's say it's just virus X is SARS-CoV-2, and then we go to influenza, and we get samples that are validated, you know, that they're flu A, and we take a lot of images of them, and we end up with all these different bounding boxes that then we can feed into our convolutional neural network, which then will give us a trained model, the trained convolutional neural network. So now we get a new sample. We start essentially using it as a test to see if a patient has either of the two or actually both. And we are able to get a result whether they have the virus, the SARS-CoV-2 or the influenza, and accordingly. So an easy way to essentially present your results in machine learning is through what is called a confusion matrix, and that's just show you the number of positives against the numbers of. So right, let's just start again. So we have a target class, so that's our ground truth. So we know, for example, that this is SARS-CoV-2, and then we compare it to what the machine learning, what the trained model tells us it is. And that's used as a metric to be able to find the true positives, the false negatives, and so on. And from that, we can derive the sensitivity and the specificity. So when we talk about sensitivity and specificity, we talk about the number of true positives divided by the true positives plus the false negatives. And I have also the other definitions for specificity: positive predictive value, negative predictive value, and the overall accuracy of the trained model. So as it should make sense, the higher these stats are, these metrics are, the better your model is performing, and the more confident you can be in your results. It's also worth mentioning at this point that the sensitivity and the specificity I'm talking about is not per sample, but actually per individual virus particle. So to begin with, we started working with lab-grown and lab-adapted strains, more specifically Udon, which is an H3N2 flu A strain, and IBV, which is which stands for infectious bronchitis virus, which is a type of coronavirus. And the negative I used for all those experiments were the media the virus was actually grown in. So from this, we can see that the overall accuracy for being able to distinguish a coronavirus from a negative sample is 91.9%, and that's for individual signals, for individual virus particles. So imagine you get a signal, then you can tell if that particular signal is a virus or not. In a typical sample, there are hundreds, if not tens of thousands of virus particles. So the more particles you have present in your sample, the more confident you can be in your results. And essentially, by establishing a threshold, you can achieve any type of accuracy you want. We also can see the confusion matrix for Udon, which is a flu A against strain against a coronavirus, and we can now see that not only we can distinguish between a positive and a negative sample, whether the virus is present or not, but you can also tell the difference between different viruses, which is very exciting. And other tests, in more conventional tests, you have to test specifically for one of the viruses and then for the other virus as well. So this can do them both at the same time. And then you go to the clinical samples, where we have been collaborating with the microbiology lab in the John Radcliffe Hospital, which is also part of the university, but also the NHS. And there we had the opportunity to start working with real clinical samples. We've been working with 25 clinical samples. Some of them were flu A. Some of them were human coronaviruses, such as OC43, HKU1, and L63, which are all low pathogenic coronaviruses, and of course, SARS-CoV-2. So these results sort of verified that the proof of principle got in the lab with lab-grown strains is also holds for clinical samples. We get 86.5% overall validation accuracy for flu vs the negative. And an interesting result is that we can also tell apart SARS-CoV-2 from other human coronaviruses. These are the same viruses but different strains, essentially same type of virus but different strain, albeit with lower overall validation accuracy, but this is without any optimization. And it's worth mentioning that when we first started developing these methods, we work exclusively for flu A, and we can see that it does work much better for flu than for SARS-CoV-2, but with some optimization, we are confident that we can reach the same levels of accuracy. So how are we going to do that? What's the future work? So as I've said, so far we have processed 25 clinical samples to get the sensitivity and the specificity per virus particle. What we need to do right now is process more clinical samples. We are hoping to process about 100 positives and 150 negatives. That not only will give us a better understanding of the sensitivity and specificity per sample, but also will improve our results as the more data we are able to supply to the networks, the more confident we will become to its prediction. We're also looking at optimizing our assay for SARS-CoV-2. Maybe that means that we need to use some different DNAs that essentially the length of the DNA and can affect how effective the labeling of the virus is. So there's some experimentation to be done there. And also you're pursuing some other collaborations, some very exciting ones, to explore alternative imaging platforms. So something really exciting, at least for me, is smartphone-enabled microscopy. So we have collaborators in Germany that have developed this amazing prototype that can take super resolution images of biological samples. And we're exploring the option whether we'll be able to replicate our results using their prototype. Essentially, all you need is some 3D printed parts and a smartphone, and you can detect viruses, which could be really exciting and really helpful, especially for low-income and middle-income countries. So, as some of you may have seen, there's been a preprint published last October in medRxiv, which gathered a significant amount of attention from the media. And the paper is now currently under review in a high-impact journal. We are hoping to get it published by early next year. We also have some exciting news in terms of commercialization and how this assay can actually be deployed to help during this pandemic. So there's a spin-out coming soon in collaboration with Oxford University Innovation. And the aim of the company is to bring to the market a fully autonomous device that can perform the test without the need of a lab within the next 3 to 6 months. This is ambitious, but we have done our homework and talked to relevant people in the industry, and we are confident that we can deliver this, subject of course to investment and support. So how would a commercial test work? So the aim would be to tap into the existing throat swab infrastructure. So nothing changes there. And then we are going to manufacture a device that will be able to carry out the test autonomously. So there will be no need for a lab and no need for trained people to carry out the test. Then this device, which would be connected either to a local computer or the cloud, and will communicate the results of the test, and most probably via an app. So every sample will have a unique QR or barcode, so it's linked to the individual. That will make it easier to track. So the result, in a sense, is digitized, and then potentially this can be used as a day pass to give access either to airports or venues where people socialize or work. So the results will still be delivered within 1 to 5 minutes. So it's extremely rapid, more rapid than anything that currently exists in the market. If the result is positive, then unlike other tests where they just give a positive result for a specific virus, we'll be able to tell which specific virus it is. And then according to the result, there may be some referral, or the individual may have to self-isolate. If the results are negative, then we can ensure a virus-free environment, and probably this is, at least in my head, a way to go back to how life used to be, a return to normality. So what are the unique features of our methods? Well, it's not just rapid. So other tests are rapid and they mean 30 minutes, 15 minutes. We are near real time. We need 1 to 5 minutes, and with optimization and using industry standards, we can probably bring it down more towards the 1 minute every single time. We are direct. We're looking at individual virus particles. So it's a very quantitative method. It is an antigen-based test, so it can tell you whether you have the infection now, if you're spreading it right now. And it is also multi-use. So most of the lateral flow assays, these rapid tests, they're single-use. But this is not the case with our method. So we envision that this device can be used many hundreds times, many thousands of times, and not, yeah, so we can use it multiple times. It will also anticipate it to be very cost effective. Part of the reason is because there's no laboratory required, but also have cost-effective reagents, essentially being some DNA and calcium chloride, and you don't directly compete with other tests such as RT-PCR. Our result is also digitized. You can have a digital result that can give us real-time information about the spread of the disease that can help immensely policymakers to make their decisions, but also can be linked to other digital technologies and other platforms. So some acknowledgements, of course, my supervisors, Assistant Professor Nicole Robb at the University of Warwick, and Achilles Kapanidis. I couldn't have done this work without them. Last year, I had the opportunity, and I'm extremely grateful to Alex Tometsky. He's the second author in the paper. His help has been invaluable, especially with the machine learning side of things. And the other virus people in the group, Christoph and Andrew, and the rest of the Kapanidis lab. And of course, my graphics card, which has been doing most of the hard work. We also been lucky enough to be funded by a wide range of bodies, including BBSRC, the University of Oxford COVID-19 Rapid Response Grant, and I am personally funded by UKRI and more specifically BBSRC through the Interdisciplinary Biosciences DTP, and I also have a scholarship from the Levendis Foundation, so I'm really grateful to these funding bodies. And thank you for listening.
Thank you for the fascinating talk. We have several questions coming from the audience. May we have the first one on the screen?
Okay, probably it would be easier if I just stop sharing my presentation, right? So let me do that
Yep,
first.
I
Okay.
can always
So
read it.
shall I just go and?
Well, the question goes: is the calcium
Yeah, sure.
chloride
I don't mind.
if you're using this technique to detect, say, SARS-CoV-2 infected viral cells, which I'm assuming is a cell line, how do you control for unspecific labeling of these cells?
So part of the thing I haven't covered is how we stick the virus. We use a biopolymer called chitosan. So this is really good at sticking small stuff, but not very good at sticking big stuff. So essentially, it does label other kinds of cells, but we never see them on the surface. And that's part of the reason why I use TIRF, total internal reflection microscopy. And it is not specific, it's unspecific. So it labels anything with a membrane. That's the way it works.
So would it also label membranous organelles, say, in the whole cell, or?
So it does. You can see the images, but these are huge. So if by any chance they do stick to the surface, which it never does, it will be discarded by the pre-processing steps where it filters by size. So a virus is about 100 nanometers in diameter. A cell is really big. It can be several micrometers, hundreds of micrometers. So the difference in size is huge.
So it's rather good at discriminating viruses. Right. I think that's a good answer to the question. May we have the second question, please? Is the neural network able to reliably reject non-SARS-CoV-2 viruses that it was not trained on?
So essentially what you do with supervised learning, you just tell it, you give it two options and you tell it has to be one of these two options. So it's really hard to predict what it would be. So there are methods that can be deployed to make sure it doesn't confuse it for other viruses. But just to be safe, there is only a limited amount of pathogens, and in this case, viruses that are circulating in the population, and that's the seasonal flu, the seasonal coronaviruses, and in this case, the SARS-CoV-2. And as we've shown from the results, it is really easy to distinguish between these three broad categories. So there is no reason not to train the network on everything, if that makes sense. I wouldn't risk it.
And it actually links in really well with another question we've had. This question goes, so theoretically, is it possible to detect an unlimited number of different types of viruses in the same sample? And this actually came from one of our audiences, and Andrew.
So I think that would be really hard. Essentially, the more different viruses you give to your network. So the biggest number I tried was 5 different classes, so 1 negative and then 4 different viruses, and you can distinguish them with about 85% accuracy. So the reason I wouldn't go further than that, for example, 10, 20, infinite number of classes, is you wouldn't be able to trust the results. You wouldn't be able to differentiate them, nitpick, because essentially it tries to find something that differentiates the particular virus from everything else. And that would be really hard in a scenario where you have that many viruses. So all the features essentially are averaged out and you're not able to differentiate them effectively.
Okay, I see. But because there will only be a few strains of viruses in circulation and population, it actually there will be no need to screen for example, more than 5, which means you can get resolution.
So when I say 5, for example, flu A, that can actually be many different strains. It's just because they belong to the same type, that's fine. So you can distinguish types easily. And even if you want to narrow down on a particular strain, you can also do that as well. It's just when you just start increasing the number of different classes and viruses, the unique features, because we're talking about diffracted limited images, they're not very information heavy. So you start averaging what's important, because viruses share different they're not. They have similar sizes and so on. So I wouldn't try more than, let's say, 5.
That's a reasonable estimate, I say.
Yeah.
May we have the next question? Does this technique require less skilled labor than the methods you discussed at the beginning? I suppose we've kind of answered the question.
Yeah, so for example, you can deploy fluidics, so things can happen by itself, but also something like what DNA Nudge does, which essentially is another SARS-CoV-2 test where they make use of a capsule. You add your swab in, then this is biocontained, and then you can just use it straight for imaging. So you can do something similar to that.
Right. Okay, next question, please. Next. What's the real-world cost of this method? Can it be readily distributed and used in developing countries or infrastructure-poor regions of the world? And it's from Lucas.
Yeah, so one of the social mission of the company is actually to create an affordable platform to detect viruses in low-income and middle-income countries, and we are confident that we can deliver this. It's going to be substantially cheaper than current tests, that's for sure, and that's partly because of the university's policies to make readily available any SARS-CoV-2 technology to the world at no cost or very low cost. So it will have to be cheap. And when we say cheap, we're not talking about hundreds of pounds. We're talking about tenths, like 10, 20, 30, 40, I don't want to say a specific number of pounds.
Per test or?
Per test, yeah. So for RT-PCR right now, is over 100 pounds per test.
Oh, wow. Okay. So I think that puts it.
Yeah, and when you scale it up, it also gets cheaper as well. So
that's very true. And I suppose a lot of this, the process can be carried out remotely, I suppose. So for example, the machine learning part, you can just presumably upload images onto the internet, so you won't have to exactly have the model.
Exactly, and that would be very helpful in a scenario where in a low-income countries, as Lucas' question was referring to. And we're even exploring options, like I said, with some collaborations on doing these things on a smartphone. Yes. And everyone has a smartphone, so that would be super easy.
Indeed. Could we have the next question, please? Is there any chance of this method being used on non-enveloped viruses?
So I have tried this method, although it's not. So on different, with different labeling methods. So essentially, to make the jump to non-enveloped viruses, we'll have to find another labeling methodology, which they exist. They're just not as fast and as reliable as the one my lab has invented. So technically, it is possible. It's just bringing it down to a sensible time and making sure it's robust and it's affordable and cheap. It's not just what is possible, is whether it can be mass produced and it's suitable to be mass produced, if that makes sense.
So how did you guys come upon the calcium chloride solution for the envelope virus? Was it mass testing, or was it just?
So it's like an anecdote in the lab. So they were just trying to replicate some results of a paper. I don't even remember the title. So that was using aptamers, which are essentially pieces of RNA or DNA that bind specifically to a target. And they couldn't replicate the results. And for some reason, the paper suggested really high concentrations of calcium. And one of the controls, they didn't have the calcium, it didn't work. So it was sort of like a happy accident, if you will.
So you chanced upon that calcium chloride just happens to.
Yeah, that was not done by me. That was before me. I was lucky enough to join the lab after the labeling method was discovered. Yeah.
Okay. Well, you see, there's always coincidences and good luck everywhere.
Indeed.
May I have a next question? Will the method still work if the virus mutates? So, because you said that the probe or the calcium chloride actually sticks to the envelope proteins, which presumably could change if the virus mutates.
So it sticks to the membrane, not the proteins on the surface. But certainly, if something changes on the surface, it will have an effect because there are lots of different interactions between the different parts. So presumably, it depends the extent of the mutation, what mutates, but also when we do clinical samples, if essentially it's trained on them, if a mutation currently is circulating, because we are training on it and because we are always able to save the data that we go through, if we perform a test, there is some data that it gets stored, we can always retrain and go back and improve our results. So if there is a mutation, so we've actually tried exactly that. So go for a particular strain that is just a different mutation. And we've done this for H3N2 with X31 virus and Udon, and it is still able to tell them apart with less accuracy, but still is able to tell them apart, about 75%.
Okay.
And then with
So
a lot more, the sensitivity increases. Yeah.
So how often do you reckon you have to retrain this model based on, because viruses constantly mutates? I am a biologist, and that's what we get taught.
So essentially, for seasonal flu, so flu mutates very often, sort of like every year, and that's why we have the flu jab every year. It will need to be done once a year, but that's only if you want to tell, not if there is flu A VS negative, but also if you want to know exactly which strain it is, which is possible to do with this method. So it has to be done once every year. Once you have the device, it's easy to retrain. You just have the same device. You just get some samples and you train your network, and that's about it. With coronaviruses, they don't tend to mutate that often, so at least not significantly. So I wouldn't. It depends for the case. For example, the low pathogenics, I wouldn't expect them to mutate in the near future.
Would it matter that maybe in a different region of the world? Because you just mentioned that you think that this technology will be applicable to various parts of the world. What if they have different forms of the virus? Would you have to retrain the model, or?
So I think it's a good thing to do. So if we go in a country, so that will depend on the type of sample. For example, if it's a saliva sample, if it's a throat swab, potentially there are going to be some differences in different countries, maybe because of what people eat. So for example, if I'm from Cyprus, we eat lots of like Mediterranean food. If our saliva may contain different things that what we're used to here in the UK, and that may mess up with the labeling. That's just an unknown that we have to test before we deploy the test. Just to give you an idea, though, just to gather a training set requires about 1.5 days, and you don't need to retrain until the virus changes. So 1.5 days is not a big deal, if you ask me, just to have, peace of mind.
Yeah, I think that is pretty amazing given this test is so cheap and so fast. Right, I think we have one more question coming from the audience, and that'll be the last one. In terms of portability, what sort of computing requirements and hardware do the imaging and AI staff require?
So if we're talking to device manufacturers, and essentially what we're using right now is a very high spec microscope, but we're confident that we can strip this down to the bare minimum because we're not actually using something that fancy. So we expect that to be quite cheap. It has to be portable, and the computing requirements, the only computational power you really need is during the training, but that can be done remotely, for example, on a cloud. So at the end of the day, you may just need your smartphone.
Okay, I think that concludes the talk. I'm afraid that's all the questions we have. Again, thank you so much for your time, Nicholas. Given the topical nature of your talk, I'm sure many of our audiences enjoyed it as much as I did. Thank you to all those who tuned in today. We have had some brilliant questions coming from the audience. As a forecast, this Thursday, The last talk of this term will be given by Dr. Rollier. Dr. Rollier is a member of the Oxford Vaccine Group, and she'll be talking about technologies behind the COVID vaccine development. We hope to see you there. Have a lovely evening.